Hello and welcome to this online tutorial about ViTa, or the viral microRNA targeting tool used for the prediction of host microRNAs onto viral sequences. During this tutorial, the following points will be covered: the microRNA basics, what is ViTa about, what is examined by ViTa, what are the algorithms, and what is a web interface like. However, the web interface and its interactions will be explained via a research question to give more of a biological background to it and a clearer understanding to what it can really do, followed by a discussion of the pros and cons of ViTa and an elaboration of questions that might occur during the presentation. Now, what is microRNA? To start off, we take a closer look at how microRNA is formed. MicroRNA is formed by transcribing DNA with RNA polymerase II. This results in the creation of a primary microRNA fragment which will fold in its signature loop 3D loop structure as identified here. However, this precursor will be cut off in order to form the pre-microRNA molecule. It is cut off by the microprecursor complex, resulting in the formation of pre-microRNA. Pre-microRNA is then recognized by the export protein exportin 5 and transported out of the core via a nuclear pore into the cytoplasm. Once arrived in the cytoplasm, exportin 5 and pre-microRNA will shed, following the reuptake of exportin 5 into the core. Soon after, the pre-microRNA fragment is recognized by dicer, which will cleave off the loop structures as identified here and here. This is now a mature microRNA molecule. This mature microRNA molecule is then taken up by an organoid protein, in this case AGO2. AGO2 will then unwind the 3D conformation into a 2D conformation, following the separation of the guiding strand from the passenger strand. The passenger strand is dissociated while the guiding strand will remain in the AGO2 molecule. AGO2 will then bind to other organoid proteins to form the RNA-induced silencing complex. This complex can now bind to a messenger RNA fragment of interest in order to prevent gene expression and proteins to be formed. Do take note of some remarks. For instance, the complementarity between microRNA and messenger RNA is higher in plants than in animals, meaning that plants have almost 100% identical residues, while in animals this often isn't the case. This will be reapprehended in the tool demonstration. It will also be noted that there are either good and bad effects to gene silencing. A good effect might be the repression of viral proteins from being formed, while a malignant effect could be the silencing of an apoptosis gene for a certain cell family. This can result in cancer and thus the uncontrolled proliferation of cells. However, a good and bad dual character might be possible for microRNAs, but this will be apprehended in a few slides from now on. Now, a bit more information will be given about the online tool for viral microRNA targeting, or ViTa. In order for the following information to be accurate, a, a reliable resource on the ViTa tool was accessed, namely the current displayed article ViTa: prediction of host microRNA targets on viruses from the Nucleic Acids Research Database. The ViTa tool is an online prediction tool that works through a web interface. It aligns the host-generated microRNAs on possible viral sequences. Do know that this is a prediction tool and this may not always be what is occurrent in a real-life situation. Thus, the in silico approach may vary from in vitro and in vivo approaches. ViTa tool itself comprises of several resources. For instance, viral genomes are acquisitions from CBI Databank and the International Committee for Taxonomy of Viruses or the ICTV Databank. Viral targets are processed from literature, while host microRNAs are derived from the microRNA base or database. An experimental proof is gathered through microRNA map. The sequences have been entered. ViTa is able to visualize data in an enormous array, as presented on the slide itself. Although I want to point out something interesting that I hinted to in a few previous slides regarding the dual character of microRNA. For instance, in this slide from microRNA map, there is a bead array experiment representative of a certain microRNA strand. Though it can be seen that there is an upregulation in the normal uterus and a slight upregulation for the same tissue when it is related to carcinogenic formations. So this might indicate that the same microRNA is, while not being fully expressed like in normal uterus tissue, it is still present in tumorigenic tissue, which might identify that there is a good and malignant side to the microRNA used. Other than this, experimental data is also gathered in qPCR format, which is not shown here. Onwards with the algorithms used by ViTa or the viral targeting microRNA targeting tool. ViTa comprises of two main algorithms, target scan and Miranda, which have both a similar type of approach. But let's get a closer look at it, shall we? The Miranda algorithm applies a similar score, the Miranda score in this case, just like a Swift-Waterman alignment would do in BLAST. Of course, this is similar to Swift-Waterman as we are dealing with a small nucleic sequence, and Swift-Waterman is for local alignment, so it makes sense in that case. But just to clarify myself, there you go. In this very case, for GC pairs and adenine uracil pairs, a 5+ is accounted for, while there is allowance for wobbly pairs to be formed, such as guanine with uracil, and a +2 is accounted for. This is quite important as it is a key component to identify duplex formation on RNA-RNA basis. A mismatch is deducted with 2 points, while gaps are deducted with more severe points. That is, for an opening there is a deduction of 8 points, while an extension is only -2 points. Besides this, there is some additional information that has to be fulfilled. For instance, the first 11 positions, negative or positive, are multiplied by a scaling factor which can be modified through the Miranda algorithm. Apart from this, the entire alignment has to be in sync with 4 empirical rules starting at the 5' end of the microRNA molecule. For instance, mismatches at the 2nd to the 4th nucleotide aren't allowed. Neither are more than 5 mismatches allowed ranging between nucleotides 3 to 12. However, 1 or more mismatches are allowed between nucleotide 9 and 5 ranging over the entire length of the microRNA fragment. And only less than 2 mismatches are allowed in the last 5 positions of the microRNA fragment. Once these empirical rules have been fulfilled, a proper Miranda score can be accredited to the given alignment. Target scan is also an algorithm that works based on alignment. However, for target scan to work, the first step is for the microRNA fragment to find an identical array of nucleotides to the corresponding untranslated region of the messenger RNA strand. This happens between the second and the eighth nucleotides of the microRNA fragment. These have to be filled in completely without any gaps in order for the first step to succeed. Once the seeding match is established, the extension of both ends can occur. The extension will occur till a gap is encountered, like here the first nucleotides stop, and here the other nucleotides are aligned until a gap with no identical residues is identified. Other matches occurring at the 3' end of the microRNA fragment are then formed and aligned with each other for the entire alignment to be optimized. Once fully optimized, a Gibbs free energy score is assigned to every individual match. Once this value has been assigned, the alignment is further scored. Then the process is repeated for other organisms with similar UTRs, but in the example we are going to process, this will not be required. This slide provides a general overview of how the ViTa algorithm works. So data is acquisition, then data is assembled, and then, which might be a bit more clearer than first shown, is the target prediction. So Miranda and targets can both yield a score based on the likelihood of the residual alignment, and an MFE value, or a minimum free energy value, is assigned. The lower this value, the more probable hybridization is to occur between the two RNA sites. Finally, we get to the good stuff, the research question and the elaboration and the examination of the tool itself. Thus, via this example, I want to apprehend the Sudan Ebola virus. I would like to examine if there are microRNAs present within the human body that target this specific virus in order to minimize the viral reproduction, and this by targeting one or more different areas of the coding sequence, because if you don't target the coding sequence, there won't be any good use to it, will there? Now let's get on with the real stuff. How does Ebola enter the cytoplasm? Well, after a lot of trial and error, it is able to engulf itself into into the cytosol with a lysosome surrounding it. So this engulfs itself, and then the Ebola virus finds itself present in the lysosome. So in a lysosome, there are a lot of enzymes present which will tear the virus apart. So it wants to get out. The way it does this is through the signaling molecule NPC1. And NPC1 sends out chemical signals to the virus for it to bind to it, because it has evolved to bind to this specific channel. And once it binds to it, it is able to release its viral load. The virus binds with these channels through the usage of glycoproteins, and research on NCBI has revealed the coding sequence shown right here for these glycoproteins. Now what we want to do is target with human microRNAs the specific areas of this CDS for it to prevent coding the glycoproteins on the outer shell, resulting in the fact that it can't interact anymore with NPC1, resulting in a dissemination of the virus within the lysosome. But let us get working now with ViTa through the website itself. Once you have navigated yourself to the ViTa homepage, you will enter the virus of interest. In this case, it is an Ebola virus. Go and press go. We want to look. In this case, we are looking for the Ebola strain, Bilovirus strain Gulu, and a complete genome, which is quite good because if one of these microRNAs are targeting several genes, we'll get a lovely overview of those. Then, what you can see is some basic information here. So the accession number is given, the total length of the genome. This is a complete genome, and what type it is. So single-stranded linear RNA, type of organism. Then some more information. And here we can view all of the microRNAs, and here you can show more in a separate window if you like to. But we will focus on these presented in the lovely map view here. So we want to so this between the 5998 and 8027, so this region right here, and we will just select one that might look interesting enough. So let's go for this little brown one, and here we have our target location. So our target location is specified here, and the virus gene is unknown, but thankfully to previous research we know that this is a region coding for the glycoproteins. So here we have a minimum free energy of -15 and a Miranda score of 156. And here we have the hybridization given. Do you know that this hybridization is the hybridization between the um, between the between the guiding strand and the passenger strand right before it is separated by the Argonaut protein 2. So that's the hybridization given right here. If we click on Miranda right here, we open it in a new tab, you will get a map with the current alignment present. So let's scroll a little bit down, and here we have where it binds to. So it's quite interesting to note, and here we have already a wobbly pair alignment. That's quite good to note at first rate. Here we have no alignment, no, 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 and here we have again alignment, again, again. So this might be the seeding region actually. Yeah, it might form quite a good seeding region based on three nucleotides right there. Then we have no attachment, and then yeah, that'll be it really. So as noted again, in animal sequences there is nearly no alignment for the target of interest, or a very low alignment percentage as earlier apprehended between the plants and the animal kingdom. And if we press target scan, we can get a closer look at the predicted area for target scan. It often is only 8 to 7 base pairs long, where we can get some hybridization information. So this is the actual strand of 35 nucleotides long of the UTR, and this is then again of our gene of interest actually, and this is the microRNA binding to it. So here we have some attachment, so probably pair here, nothing quite of interest here. Oh, here we have a perfect alignment. Yeah, it should be better if there was a more clear alignment present right here. But what we can do is we can always select the microRNA itself, and it will present us with a page of the microRNA mapping. So here we have the species Homo sapiens and description what it is. It is found on chromosome 5. It is intergenic and the pre-length is 88 nucleotides. So here we have the red area is the entire parent strand, or the guiding strand of our pre-microRNA fragment. And here it says this microRNA sequence was predicted based on homology verified by microRNA from mice. Expression of in human demonstrated significantly reduced levels of microRNA in precancerous and neoplastic colorectal tissues. So that's quite interesting to note as well. And the beta ray experiment will tell us either. So beta ray experiment and qPCR results will tell us where it's upregulated. So here we have the colorectal and of course the prostate gland near there, and the bladder all in the same area, and the cervix for females, and the ovary just all in the same area. Quite interesting to know, but very much for prostate. And here we will know that the microRNA is also current in, as noted by the comments right here, demonstrates significantly reduced levels of microRNA in precancerous tissue, but we see upregulation So pre-cancerous tissue and then cancerous tissue we see upregulation. So this might be a lovely example of the dual character of microRNA. So it is present in regular samples, but it is also present during cancer, which shows the good and the malignant properties that this microRNA can have on a human being, which might not be very wise to implement this very microRNA target. So we'll have to look for another microRNA target that might be more fitting to our needs. But this was a general quick look to what ViTa has to offer and what it really does. What you also can look at, if you don't think this information is too clarifying, ViTa has a database of its own. So if you go to home, because this is microRNA map, and you go to tissue specificity of microRNAs, do note that this is a database of 2006 and it might not have been updated. So we want to look for 145. So let's quickly look it up. 145. Oh, and there we see it. Yeah. All right, microRNA 145. But the thing is, the upper bar doesn't quite come along. So we want to look at colon again, and this is tumor and this is normal tissue. So we can have a look at those. Come on, show. And here we see in normal tissue it's upregulated and a little lighter expression in tumor tissue. So that's quite interesting to note as well. So you can look it up in both directions, but you can see here that there is a lot of unknown data, a lot of data that is still missing. So it would be lovely if this would be updated. But I hope this provides you with a little more information about what ViTa can do. So about ViTa, there are some good sides to it and some less good sides about it. So good side is the broad aspect of the tool, along with the interaction with several databases, as we saw in the demonstration. There is quite a lot of interaction, and the data provided is clear and nice, although it lacks a little bit in updating information. For instance, the expression profiles of ViTa itself, all the gray areas. God, stand by me, but that wasn't nice, was it? Furthermore, viral information and viral gene data when a microRNA is selected to what gene it applies and all those things, just the small things, but it could make the tool so much more powerful than having to research it on other separate engines like NCBI itself. Furthermore, clear alignment overview is what lacks as well. When you press Miranda, you only see a red and green bluish alignment, but not with a clear identification of the seeding area or possible wobbly pairs or good alignment pairs. That isn't visualized, and that should definitely be implemented. And perhaps a scoring matrix like NCBI has to offer for identities. When you have so much percentage identity, this is most likely a good alignment and so on. So if it would be better at predicting those things and implementing a similar scoring matrix for identities, it would make the tool so much more powerful. Anyhow, due to time constraints, I won't be able to go to questions. So I hope you enjoyed this tutorial. Thank you for watching, and if you have any questions, leave them down in the comments. Bye bye.